Well, Dr. Swadan, Dr. Rangaraj, a pleasure having you here to be able to talk a little bit about your project. Um, my name's Keith. I am with the Cure Starts Now. I'm co-founder of the Cure Starts Now and the DIPG DMG Collaborative. And we're excited to be able to talk a little bit about your grant on evaluating 177LU, which is delivered by CED for DMG with radiation therapy. Has a much larger title, but I'll spare you from that. But it's a phase one clinical trial. Before we go ahead and jump into the details about this trial and learn a little bit more about it, though, I wanted to share with you some good news. On behalf of the DIPG DMG Collaborative, I'm pleased to let you know that they are awarding a grant for $50,000 to start the project and for the initial aims of your trial.
That's fantastic news. Thank you for starting with that. I
Well,
appreciate.
Well, and with that, I have some folks here that also wanted to introduce themselves too and offer congratulations. Jenny, do you want to go ahead?
Before you
Yes, sir.
before you pass the mic, you were very humble in your introduction. And for those of you who don't know, and I think that's only Rashi, you introduced yourself as such, but you've been so influential in moving the platform of translational research in DMG. And I don't remember the first time we got introduced at one of the cooperative group meetings, but the concept of working together and collaboratively was presented, and that's gone from concept to very meaningful discovery and actionable items. So congrats to you, the group, the collaborative for being so instrumental in keeping DMG front and center.
No, and it's an international effort, and I think
I
that's
know.
what you're going to see in this call with it as well. I mean, it's now in I think we're in five, six countries worldwide. We have effectively 86 partners between chapters and collaborative members, and so it's a big, big effort
Yep.
that hopefully can come through and try to support more seamlessly some of the great work that you're doing. I know
Yeah.
that. Dr. Swinan, my goodness, I think we met in 2009, and obviously talked a little about this whole idea of focusing on these cancers and getting to the point that we have informed clinical trials. Well,
Yep.
that's what this is.
Yeah.
So it's quite humbling to see that. So, well,
Yep.
thank you.
Note of congratulations. That's all.
We appreciate it, and we're here to talk about you guys, though. With that, Jenny, do you want to provide a congratulations and introduction?
Sure, absolutely. My name is Ginny Pritchett. My husband, David, and I started the Virginia chapter of The Cure Starts Now. We were one of the very first two chapters. We started it back in 2009 in honor and memory of our daughter, Morgan, who battled the IPG for 20 months. Um, so I've been around for a while now, and I agree. It's been amazing to see the progress that's been made, and happy to be here today to congratulate you all on your grant and say thank you for the exciting work that you're doing. Um, and now I'd like to pass it over to Martin.
Hello, my name is Martin Chovanec. I'm from Prague, from Czech Republic, from Europe. I'm very happy that I can congratulate to you and your whole teams that you have applied for this grant and that you were awarded, and you can do this very interesting diagnostic approach for the IPG. We represent the chapter that is related to our daughter Nina, who battled with the IPG for 4 months, and I really hear that it's fantastic work that you are doing, and we keep the fingers that you will succeed with this preliminary work on management with diagnostic approach. So let me give the chance to Stacy to talk with you.
Thank you, Martin.
Hi, I'm Stacy Clark. It's nice to meet you guys, and I'm so excited to be here. I started the chapter Kentucky Beckham's Bloodline back last year, at the beginning of last year, in honor of my grandson Beckham. He fought DMG for 23 months. Um, he passed away in July. So seeing all of this progress being made with the research and being able to be a part of the grants being presented has really meant a lot to me and my family, and it's kind of helping us get through, the loss that we've had. So I'm really excited to see what you guys are going to do with this.
Thanks for sharing,
And I'm
Stace.
going to hand this over to Rachel.
Hello, my name is Rachel, and I'm the Research Support Coordinator at the Cure Starts Now. Congratulations on your grant. We're very excited for you and excited to see the results of your research. And just one question for those here and for our listening audience. What gap in current DMG treatment options does your study aim to address?
So I can start with that if we're done with the intros. So I don't know how familiar the group is with our past work with direct administration of radio-labeled monoclonal antibody for DMG, specifically in the brainstem. That was a monumental task from the standpoint of demonstrating safety, concept of instilling drugs directly into the brain. So we're not the first to do that. Others have done that. But what we did is with a very refined dosimetry map. So the fact that we're using radio-labeled antibodies gives us a great capacity to look at very important, we think, very important features such as distribution, the time scale of readministration of drug. So we can follow these patients with PET imaging longitudinally to look at tumor coverage and then start to refine things like scheduling. This particular grant, this particular project, I should say, is really following on the coattails of that with a much different platform. It's a different radioisotope for various reasons. Cost containment is a big part of that. So we're ratcheting down big time from the standpoint of the dose cost, per patient. Number one. But what we hope to really demonstrate, we're holding our feet to the fire from a phase 2 idea. Yes, there's a phase 1 component, but this will be the first time we go after the tangible outcome of basically measured outcome from the standpoint of survival. So we will be, filling the gap from the standpoint of repeat dosimetry. So repeated doses based on dosimetry maps, as well as volumetric targets in the tumor itself. So our phase 1 study was really a sampling of variable tumor sizes, variable drug distributions, variable doses, but it's our intent to rapidly but satisfy the phase 1 component given the different isotope, and then dovetail that into a phase 2 dosimetry-based repeated drug administration platform. That's a little roundabout way of answering your question, but hopefully you can appreciate the goals here.